I'm a biology major at Vassar College, and today I'm going to be telling you about the population genetics of coyote willow, or Salix sicula, in the riparian Middle Rio Grande ecosystem. And my mentor for my project this summer was Brian Alfaro, who works at the University of New Mexico. So some of you may not know that we actually have a riparian ecosystem here at the Sevilleta National Wildlife Refuge. And what a riparian ecosystem is an ecosystem surrounding a body of water such as a river or stream. And here we have a riparian ecosystem surrounding Rio Grande. And they're very interesting areas to study because they have a high level of species biodiversity. And what was interesting to me as a population geneticist is one of the most important mechanisms of seed dispersal for plants in a riparian ecosystem is seed dispersal via water, also known as hydrochory. Oops, sorry. Hydrochory, once again, is the dispersal of seeds and other plant populations such as clones via water. A really great example of this is the coconut, which is transported by ocean currents throughout different tropical islands in the areas where it's found. And hydrochory is very important in riparian ecosystems because it's the main mechanism by which gene flow occurs. Gene flow is the movement of genotypes within and among populations, often as a result of migration. And this is very important in the study of evolution because it's one of the main driving factors of evolution in the long term. The study system in which I was examining the effect of hydrochory on gene flow this summer was the coyote willow, scientific name Salix sicula. This is a native plant, a member of the willow family. It's a shrub-like willow found in wetlands and spring banks throughout the western U.S. It's a very effective colonizer, and because it reproduces both sexually and clonally, it has multiple dispersal mechanisms. Three main dispersal mechanisms that it utilizes are hydrochory, wind dispersal, and clonal dispersal. To guide me in my summer research, I asked these questions. What is the genetic structure of coyote willow populations in this region? How much gene flow occurs between populations? And how strongly does hydrochory influence this gene flow? And my main prediction was that hydrochory would strongly influence genetic diversity in coyote willow populations. And I would see this in my research through high genetic diversity within populations. Linear gene flow between populations going down the waterway, greater genetic diversity within downstream populations because you have more seeds and clonal propagules traveling downstream, collecting in downstream areas, so you have sort of a larger gene bank, so to speak. And I also predicted that I would see genetic differentiation between populations along two separate waterways because I was looking at populations both along the Rio Grande River and along an artificial drainage unit created by people, and it's located about half a mile west of the Rio Grande. In order to answer my study questions, I selected 8 sites along these two waterways, along the natural waterway, the Rio Grande, and the artificial waterway, Unit 7 Extension Drainage Unit. All of these sites were located either within the Sevilleta National Wildlife Refuge or in the Eslado waterfowl management area, which is the area just north of the refuge. Here, for reference, in the top left is the Sevilleta field station where we are currently, and these are all of my different sites. And the sites were spaced out, populations were spaced out on average 1 to 1.5 kilometers apart, although there were a couple that were a little closer together. There's some diversity in the types of sites at which I collected populations. Here you can see on the far left, one of my Rio Grande population sites is located in a dry riverbed, which now probably has water in it, now that we have officially started monsoon season, sort of. And several of my populations were adjacent to this road that goes along the drainage unit. And my two southernmost populations were actually just north of the Santa Casia Dam. But the unifying factors over all of my population sites were that they contained large adult stands of coyote below. And another commonality that I noticed was a large amount of non-native invasive species. The two that I found a great deal of in particular was salt cedar and Russian olive. When collecting, I created a transect along the bank parallel to the waterway, and I collected a small leaf sample from each individual, 15 individuals per population, and each individual was spaced approximately 4 meters away from the others. And I recorded the geographic coordinates of each individual that I sampled. Then I spent the rest of the summer in the lab working with molecular methods to genotype my coyote willow samples. And one thing to note here is that the relatively recent application of these molecular protocols in seed dispersal ecology is a really great step forward. Before this, people were actually going out into the field and collecting seeds in a waterway, or actually having to physically see the dispersal taking place. But now we can use genetics to answer those questions, which is very exciting. The three major molecular protocols that I was using was DNA isolation, which I extracted DNA from leaf samples, polymerase chain reaction, or PCR, in which I amplified DNA samples so that I have a larger amount to work with, and genotyping, in which I run the PCR products on an agarose gel to examine bands of DNA bands of different lengths that correspond with different alleles. I'm going to go over each of these in a little more detail. For DNA isolation, I physically ground the leaves and then extracted and purified DNA using the CTAB protocol. I had to test different protocols to see which would be the most effective, because different specific species just can work better with different extraction protocols. CTAB worked the best. Then I amplified the DNA using the standard PCR protocol with ISSR primers to amplify the DNA. And as a reminder, I had 120 total samples, 15 individuals per population, and 8 populations in total. And I ran PCR with different primers, and I was looking for 2 to 8 successful primers. And what I mean by a successful primer is that it cuts up the DNA into pieces and bands of different length, and these bands show up on the gel. And for a successful primer, you'll see 4 to 6 clear, dark bands of different lengths that are present in some individual samples and absent in others. These are called polymorphic bands, and they're representative of genetic variation. As I said, I put the PCRs, ran them through a gel, and then I took my gel images and put them into a computer program called Gel Analyzer. This is what one of my gels looks like in Gel Analyzer. On the far right, you have the DNA ladder, which is just a reference to know what the approximate lengths of the DNA fragments are. And each column on this, to the left of the DNA ladder, is a different individual, the genotype of a different individual. And all of the white dots represent that the program has picked up a band at that spot. And as you can see, there are some bands that are consistent with the same length throughout all the individuals. And then there are some that are present in some and absent in others. And that's what I wanted to look for. Now, this is the first population genetic study of its kind using my study species, Salix decidua. And as a result of this, the majority of my time this summer was spent developing and refining a molecular protocol that would give me usable results. And because of this, I wasn't able to genotype all of the individuals that I sampled. However, I do have some preliminary results that were collected using loci, so 4 areas in which a band was either present or absent. And I had a sample size of 35 individuals or genotypes to date, 8 samples per population in 5 out of the 8 populations that I collected. I did an ANOVA test of molecular variants. It's very similar to an ANOVA test, if everyone's familiar with that, and found that 72% of the variation between coyote globe individuals occurred within populations, and only 28% of the variation occurred between different populations. However, there's a calculation called the pi-t value, which uses both of these numbers in order to quantify how genetically distinct populations are from each other. And this value was significant. So although the populations had a lot of diversity within themselves, overall they were genetically distinct from each other. And you can kind of see this more clearly on this figure, which has the frequencies of different genotypes or different combinations of polymorphic bands in each individual population and overall data set. And you can see that there's a lot of diversity within all the different populations. Each one has at least 3 different genotypes. But then there's also a lot of difference between the populations. The top left one, the teal-colored genotype is most common, whereas the dark blue is most common. And SR1, the red one's most common. The genetic variation is coming in. Another thing that I thought might be indicative of the importance of hydrochory is that the lower 2 populations seem to have more of an even spread of different genotypes, whereas the upstream populations, 50% of the individuals are one genotype. The bottom 2 have more of an even spread, and that might be indicative of seeds and clones traveling downstream or downstream. They're also quite similar to each other, which is surprising because the southernmost populations are, one of them's on the Drain, one of them's on the Rio Grande. But one factor that might have contributed to their similarity is that they are much closer together than any of the other sites, just because of the area at which I was allowed to sample. So wind dispersal might have played a more important role, and that might be a confounding factor. One last thing about the bottom left pie chart, the reason that it's so evenly in 4 quarters is because there were only 4 individuals that had visible bands on the middle. So I need to genotype the rest of those. So let's revisit my predictions at the beginning. We did see high genetic diversity within populations. We're not sure yet whether we saw linear gene flow between populations. We had such a small sample size that we're not really able to tell anything about that yet. And we did see greater genetic diversity within downstream populations. We did not see genetic differentiation between the populations along different waterways. And again, wind dispersal and the proximity of the sites might be a confounding factor. But all this preliminary data has a big asterisk next to it because the sample size is really too small. It's an incomplete data set. We need to genotype more individuals at more loci, so using more primers. We're also, my mentor and I intend to continue working on this project. I'm going to be working on it remotely with him. And we are going to try testing out different kinds of primers. And one analysis that's going to be very important for the study is spatial analysis, comparing, looking at the relationship between genetic distance and geographic distance. In the future of this project, we also want to extend the scale of the study and include populations all the way from Colorado to Las Cruces. And this way, we can not only compare populations, we can compare regions, which will be really exciting. And we'll also be able to examine the extent of wind dispersal, as well as hydrochory. I would like to thank my mentor, Brian Alfaro, Amaris Swan, John Ers, and all of the other individuals and entities on this slide. Thank you very much. If you have any questions, please bring them up.